a cell, proteins need to interact with one another to bring about a specific function like activation or inactivation of target proteins. Y2H is a molecular genetics to validate protein-protein interaction through activation of a reporter gene. In this figure, a gene and upstream activation sequence is shown. A transcription factor comprises of being a binding domain and activation domain. The DNA binding domain binds to the upstream activation sequence, and now the activation domain recruits the transcription activation machinery to activate the transcription of gene to produce mRNA, and that will translate into protein. In this two hybridization, the two domains of transcription factors are separately cloned in different plasmids. The first plasmid has binding domain used with the target protein sequence termed as PAID. The vector also has tryptophan gene to select the transformant on the tryptophan-lacking media. The second plasmid has the activation domain fused to the second target protein termed as PRAY. In this losing gene is used for transforming screening. Thus, plasmid 1 produces BD bit fusion in plasmid to produce a deep ray fusion. A genetically modified strain of yeast deficient in tryptophan and leucine is used to select successful transformant on selective media like in tryptophan and leucine without yeast cells cannot grow. Now, both plasmid and yeast are inoculated in the selective media and later plated on the solid agar plate containing leucine and tryptophan deficient media. After incubation, only transformed yeast cells containing both the plasmids will grow into the larger colonies. Inside the transformed cells, the product of the first plasmid that is fused, BD and B, comes and bind to upstream activation sequence. If the second target protein, PRAY, interacts with the first protein, then the BD and AD fragments come in close, leading to the recruitment of transcription activation complex, which brings upon expression of the reporter gene. And if both the proteins, that is BAID and PRAY, do not interact, the two domains of the transition factor, that is BD and AD, never come in close, and as a result, the expression of reporter gene not occur. Expression of reporter genes like GFP, GUS, and LAC-Z are observed under fluorescent microscope or visually. There are four limitations in this two-hybrid system. First, the largest problem is false positive. This can be caused by the ability of BAID to induce transcription without interaction with the PAID. Second, possible incorrect protein folding in yeast. Third, gene encoding target protein must be available. And lastly, fail to detect some no interactions.